I think we're ready to start, and I'd like to welcome everybody to our second half of the program this morning on our potato systemicals mini symposiums, and I think in total we have about 10 talks. And right now I'd like to welcome Asha Kub, who will be talking about some work that she's been doing, some work that she actually did for her PhD with Feng Zhao, and now she's decided to stay in on the PCN team at the University of Idaho as a postdoc. So I think we're very fortunate to have her here.
All right, so I hope everyone can hear me right now. My name is Joanna Kut, but everyone calls me Asha, so please call me Asha. As Dr. Virulence mentioned, I'm a postdoc, and we are trying to characterize plant-nematode interaction at the molecular level. And today I will be talking about one single effector from globodera pallida that is able to suppress plant immunity, and it does this by degrading some resistance-related proteins. So we heard all this scary data that PCN can cause huge yield loss in potato. And considering how big this industry is in Idaho, those numbers are really concerning. So we could ask, like, are the plants actually really defenseless against this pathogen? When the answer is yes, no, maybe, it's complicated. So how it works is, if you imagine plants, they're constantly exposed to many pathogens, including bacteria, viruses, and nematodes as well. But most of the plants, they are healthy. And they are healthy because they have very successful defense system. So what makes this interaction between nematodes and plants so special that PCN can infect potatoes such successfully? So to answer this question, we can look at this schema, which represents the cross-section of the potato root. And as you can see, the bottom part will represent susceptible plants when the infection can easily progress, while the top part represents plants that are resistant and that can stop nematodes from infecting the roots. So I won't be talking much about the infection cycle, but pretty much in susceptible plants, nematode easily enters the roots, migrates along the root, develop feeding structure, and then entire cycle ends in the female with eggs within, and those eggs can be left in the soil, and the entire cycle can repeat itself. So what happens in the resistant plants? Plants can actually detect the presence of nematode, and then they can activate some defense mechanism. And those defense mechanisms in this schema are those little pinky cells around each stage of development of nematode. So the cells, they are actually dying. They want to separate the nematode from the rest of the root, and by this prevent nematodes from further developing. So this cell death, it's called hypersensitive cell death. We heard a little bit about that in the previous sections. And what is important is that hypersensitive cell death is caused by R genes, or in other words, also R proteins. So R proteins, those are those detectors that are within those pinky cells that will detect the presence of nematode and will activate plant immunity, which is that cell death. So as an extension, susceptible plants, they either don't have those specific right R genes that will detect nematode, or somehow nematode is so successful to suppress this defense mechanism that the plant becomes susceptible. So let's have a look on the potato. We heard a great deal of talks when people try to develop resistance to globodera pallida. So currently we have only one gene, JP2, which has been mapped, cloned, and characterized. This gene is called JP2, and it confers only resistance to some isolates of JP2. And the reason I'm mentioning this because we'll use this as a tool for analyzing how nematodes infect plants. So this is about R genes. So how actually nematodes can infect the plants so successfully? Well, they have specific molecules called effectors. And I like to call effectors as weapons that nematodes use. And actually, effectors are not specific to only nematodes. They are actually produced by all types of pathogens. And those effectors are delivered to plant cells to suppress plant immunity. And that's actually what I'm working on. I'm trying to characterize effectors that are important for nematodes to overcome plant resistance. So if you think about effectors, there's actually 3 stages how we characterize effectors. First of all, there are some criteria that a given protein needs to meet in order to be considered as effector. Then we check if the given effector can actually suppress plant immunity. And the last but not least, we try to figure out how actually this effector can do that. And that last part is the most interesting. So I mentioned those criteria. There are 3 criteria. First of all, we need to know if this protein we're interested in is actually expressed during the parasitic stages. So parasitic stages, those are the stages when the nematode infect the roots. And I haven't mentioned that yet, but the specific effector I'm interested in, it's called RHAIB, and that's what I will be talking about today. So indeed, our effector, our protein, is expressed in those parasitic stages. And the second criteria is that the protein we are interested in needs to be produced within specific organs in nematodes, and those organs are called glands. And actually, we have molecular tools to localize when given protein is expressed within a nematode. So those are cross-section of the nematodes, and as you can see, there is like a black dot within the nematode that's called gland, and that's actually the spot when our protein is produced. So indeed, this protein, that's RHA1B, is produced within glands that produce effectors. And the last criteria is a signal peptide. It's actually a small fragment of the protein. It's like a tag or information for the protein that although it's produced in these glands, those small organs in the worms, it actually is going to be secreted out into plant cells to play through in the infection. The second step when you evaluate any effector is to check if this effector can suppress plant immunity. And as I mentioned before, this immunity here is that hypersensitive cell death that is activated by RHA genes. So actually, can we use these RHA genes to evaluate the plant suppression function of effector? And the answer is yes. Actually, it's really hard to work on the roots. So what we do, we express those RHA genes in the leaves. And as you can see, those circulated areas, those are the areas where RHA genes are present, and those cells, they are dying. So actually, this cell death is exactly the same cell that's like in the roots. So we have all different type of RHA genes we tested here, and among them, there is that JP2 gene I mentioned at the beginning. So in our control case, it will cause the same cell death like it would in the roots. But whenever we add this effector to the system, you can see there is no cell death. So somehow this effector can switch from the defense, from the cell death to lack of cell death, and potentially from resistance to susceptibility. And to further verify if this effector is important for nematodes parasitism, we generate transgenic potato plants that will produce this nematode effector within the plants. And then if there is pre-accumulation of this effector within the plants, and when we infect those plants with nematodes, we should see much higher infection rate. And that's actually what we saw when we count the total number of nematodes at the end of the infection cycle. So that's our transgenic plant. As you can see, there is much more nematodes than in our control case. So that additionally supports our claim that this effector promotes virulence. And the last question was to verify how actually this effector is able to suppress plant immunity. So to answer that question, we look at the structure of this effector, and there is a characteristic domain which we call ring H2. The domain is just a fragment of the protein that has very unique structure, and this structure was characterized in other proteins, so we can predict what can be the possible function of this effector. And indeed, this domain was characterized in family of the proteins called E3 ligases. They attach ubiquitin to the proteins that later are degraded. So what does this mean? Well, in the plant cells, there are proteins that are needed, and there are proteins that are no longer needed, or they are not functioning. So the plant has to have the mechanism to degrade those proteins. And that's actually this schema that represents this mechanism. So inside the cells, there is this barrel-like structure, we call this protasome. It looks like a little bit like recycling bin. So when the protein is degraded, it gets to this protasome and it gets cut into small pieces. But not every protein will get to protasome. It has to be first tagged with this small ubiquitin. And it's not like every protein gets stuck with ubiquitin. There has to be 3 enzymes, E1, E2, and E3. Those enzymes will attach ubiquitin to the proteins that's supposed to be degraded, and then the cells know that this protein actually needs to go to proteasome and get destroyed. So by this, we kind of predict that this effector may play this role, can actually decide which proteins within the cells can be degraded, which can stay untouched. And first of all, we need to verify if this effector really has this ability to add the ubiquitin tag. And those are actually experiments that we use to verify this ability of the effector to add ubiquitin. And others, I don't want you to really focus on the details. The most important thing is that when the protein has this ability, you will see this characteristic smear, which is present in both lanes. So we did these experiments both in vitro and in vivo. So indeed, this effector has this ability. So since we know that our protein is called cell death, and since we know that our effector can suppress this cell death in plants, and this effector is E3 ligase, it was an obvious question to ask if this effector actually can degrade those R proteins. So we have assay for that as well. We can actually mix those proteins together, and here we have all those different type of R genes, and here this upper panel represents the accumulation of R proteins. And the first lane is our control, so as you can see, protein accumulates in each case, but when we add effector to this, in each case this R protein is gone. And again, we included this one characterized R gene in our experiments. So taken together, what I want you to remember from this talk, effectors, those are those molecules that nematodes use as a weapons to suppress plant immunity. And the effector I'm working on can suppress plant immunity by promoting degradation of some resistant proteins. So to come back to our original schema, in the resistant plants, when nematode enter the root, it can be recognized and hypersensitive cell death can be activated, and that's how we have resistance. But then the effector, like the one I'm working on, can actually degrade the very R proteins that should detect the nematode, and by this suppress that characteristic cell death and switch from resistance to susceptibility. So that's thank you for the time, for your time, and thank you for all the people who contributed to this project, as well to finding agencies for providing financial support. And if there are any questions, I'll be happy to answer them.